If you're watching this video, you've probably just received your first analysis from data2bio and are wondering how to visualize the alignment of your sequencing data to your reference genome. I recommend that you, if possible, watch this video in full screen so you can get a better sense of what is going on. The first thing you're going to want to do is insert your flash drive and take a look at what is available in the report. So there will probably actually be far more files than you see present here in your own and the analysis you've received. I'm making this video as the report is still being finalized. For this example, the files we're interested in are this example BAM and BAM.bai, and you can see that there will be one of these sets of files for each lane of sequencing that you submitted for our analysis. In order to visualize the reads, which are stored within these files, we're going to use a program called the Integrative Genomics Viewer, which is produced by the Broad Institute. So this software is free. You can find it just by typing in igv, and you can see it is the first hit. So in order to use the igv browser, you do have to register, but this is a very quick process. And afterwards, you can log in just by entering your email address. And this takes you to a page with a number of options for the version of the Integrative Genomics Viewer that you want to launch. Ideally, you want to select the version that will use the most RAM that is available to you on your computer. Short of serious workstations, very few computers have access to 10 gigabytes of RAM. So generally you'll want to use this 2 gigabyte version, and if you're still using a 32-bit version of Windows, you'll want to use this 1.2 gigabyte version. So we're just going to click Launch, and that brings up this igv.lm file, and when we open this, you can see that now the application is being downloaded from the Broad Institute. Now this may run for a little while. So as you can see, the Integrative Genomics Viewer will initially launch with the human reference genome loaded, which is probably, there's a good chance that this isn't actually the organism you're studying. So the Broad Institute provides a number of different reference genomes that you can select from this pull-down menu. Now if you have paid for analysis of a reference genome that's not on this list, data2bio will have provided you with the files necessary to load your own. But that's a slightly more complicated process, so you'll want to see the dedicated tutorial video on that. But fortunately for us, we're interested in maize or corn, and this genome is already available. So it'll just take a little while we now load the new genome. The B73 genome has now finished loading, and we can see we've zoomed out so that all 10 chromosomes are concatenated, and we're looking at the entire genome at once, which is not a very informative view at all. So to zoom in on something we'd be more interested in, let's start by pasting in a relatively random gene name. And I'm going to actually make this a transcript ID because, as you can see here, the names of the format of the genes as they are loaded into the browser are in fact transcript IDs. So this will vary depending on what your organism is. Just make sure that you're entering your gene name in the same format as is displayed down here at the bottom. So when I do that, we zoom to the location of this gene, which happens to be on chromosome 1, about 235 megabases from the start of the chromosome. And here you can see the genome structure. We can zoom out a little, and you can see the gene and the exons of the gene. But we want to know what about our sequence data that aligns to this gene. So to do this, you can simply open a file, and we want to open this example BAM file. And when you open the example BAM file, you'll get a new track that will come in between the top and between the gene models at the bottom, with these little gray bars and gray peaks. So the top part of this track is called coverage, and it's showing you the number of reads that have aligned to each particular base, whereas the bottom part that shows the actual reads themselves. And if we zoom in on a particular read, like so, you can see that these reads will stack up and have small colored lines in them. So each colored line represents a base pair in the sequenced read which does not agree with the reference sequence, and the different colors stand for different different base pairs. So if we zoom in even more, you can see that the green is an A, and over here the brown color is a G. The other thing you'll notice is that by looking up at the top coverage track, these base pairs where there are these SNPs show the same color, and when there's disagreement between different reads as to the base pair at a specific location, this bar is actually proportional, showing the relative ratio of the two base pairs. In this case, the G, which does not agree with the reference sequence, is represented in one read, and an A, which does agree with the reference sequence, as you can see down here, is represented in the other read. So this bar is half brown and half green. If we were analyzing differential expression of genes, we could load a second file. So in this case, example 2, and this creates a second track below the first track. And as you can see in this case, there are still the same two reads aligned to the region with the same polymorphism, and that's because example 2 is actually another copy of the same file. Now if we aren't interested in this gene anymore, we can zoom out and scroll around the genome and find here's another gene. You can see that there are no reads that align to this gene, so potentially this gene is not expressed in either condition. As we continue to zoom out, you see now we're so far zoomed out we no longer would see alignments even if they were present. But we can scroll along the genome, finding new genes, for example this one, zoom back in, and as you can see this gene has many more reads aligned to it, and potentially has a SNP here, although you can see that it's only represented by one base pair, with the other three reads agreeing with the reference sequence, so this probably simply represents a sequencing error. You can also see that half of these reads are aligned here, and then there's this line leading to the other half of the reads aligned at this second exon. So these reads are spanning an intron-exon boundary. So I hope this has been a useful introduction to using the Integrated Genomics Viewer. Good luck and enjoy your data2bio results.